Welcome to NETT Day. This is number 14, I believe. My name is Mike Bedell. I'm Dean of the College of Business and Technology. NETT, of course, stands for Northeastern Empowerment Through Technology. And day is about today. So welcome to NETT Day. We hope we have an intriguing program for you, something that's forward-looking and thoughtful and grabs the leading edge. And so that's really the purpose. I want to thank the various faculty that have been involved in this, in putting this together. There's a faculty committee, Dave Jordan, who you met, Tunku, who's over here, Ahmed Khalid in the back, and some others that I'm missing at this point. But again, thank those faculty for putting this together. I hope you guys have a great day, and it's thoughtful, and do not hesitate to talk to these folks. Ask questions during, ask questions after. That's how you're going to learn. So thanks for being here. Have a great day.
So we're really excited, or I'm really excited to kind of see where AI, biology, machine language kind of intersects. And for today, it intersects right here in this next coming presentation. So really fascinating job and kind of endeavors that are going on here. So with any further ado, I'm going to invite Carla Mann to come up and give her presentation.
All right. Can y'all hear me okay? Awesome. Let me just make sure my clicker here works. It does. Fantastic. All right. So I am Carla Mann. I am an assistant computational scientist at Argonne National Laboratory. And today I'm going to talk to you guys about some of the work that our lab and lab group does using large language models and artificial intelligence to fight against both current and future pandemics. So just to kind of give you an overview of what this talk is going to cover, I'm going to give a really brief introduction to Argonne National Laboratory for those of you who aren't familiar. And then I'm going to get into the meat and potatoes. We're going to talk about AI and large language models and the actual research that I'm working on right now. Then I'm going to tell you guys how I became a computational biologist and how you can too. And I do have time budgeted at the end for questions, but if you guys have questions during my talk, throw up your hand and we can discuss them right then and there. So, Argonne National Laboratory. It's one of 17 Department of Energy national laboratories in the United States. It was actually born out of the University of Chicago's work on the Manhattan Project. So the very first self-sustaining nuclear reactor was built under the stadium seating of the original Stagg Fields at the University of Chicago. And then a bunch of people sat there thinking, hey, maybe we shouldn't build a nuclear reactor under a stadium. And so they moved it out into Waterfall Glen Forest Preserve, which is where Argonne was established in 1946, with the express purpose of taking all of that nuclear technology they'd been working on in the Manhattan Project and applying it to peaceful things and not nuclear warfare. So since 1946, we've done a lot more research, not just nuclear research, including things like energy storage, so battery technology. We do so much battery technology. Renewable energy, including hydroelectric and solar power, as well as nuclear energy. We also do just a ton of physics research in general, but we also do biology and chemistry research, and not many people know that. I grew up literally 45 minutes away from Argonne National Laboratory. I did not know they did biological research until I started working there. The other thing that Argonne does, and the really big important thing it does, is supercomputing. Argonne is one of, if not the leading supercomputing centers in the United States. We have a bunch of cool toys. Argonne, which is in this nice little aerial photo here, sits on about 2.7 square miles in the middle of a forest preserve. It's really pretty. We have deer wandering around on campus all the time. We have about 3,500 employees, including about 1,000 research scientists, all working in these fun little buildings here. We are also home to multiple user facilities, and these are really cool, large-scale scientific facilities that people from around the United States can use. So if you are in academia, if you are in industry, if you work for another government laboratory, you can come in and use these user facilities at much less in cost than if you had to build that kind of capability yourself. So some of our user facilities include the Advanced Photon Source, which is this ring thingy here. Basically, that's a particle accelerator. It allows us to shoot X-rays at really small things, and then we look at how the X-rays bounce off, and we can get a picture of what that really small thing is. So during the pandemic, if you saw any images of SARS-CoV-2 on the news or online somewhere, chances are that image was reconstructed from information that was generated from our Advanced Photon Source facility. We also have a smaller particle accelerator, the Tandem Linac Accelerator System, or ATLAS. And most importantly to this conversation, we also have this Argonne Leadership Computing Facility, or ALCF. And this is home to multiple supercomputers, including Aurora, which when it comes online at the end of this year, God willing, this time for realsies, will be one of, if not the fastest supercomputers in the world. So it's going to be capable of 2 exaflops, which means that it can do 2 quintillion math problems every single second, which is important when we are doing large language modeling and working with artificial intelligence, because those things take a lot of computing power. Can we click on the thingy? There we go. Clicker's working there. All right. I think so. The animations aren't quite working right, but we'll work with it. All right, so I'm going to talk to you guys about the work that Argonne and my lab group in particular is doing to combat pandemics. And a lot of people are paying less attention to the SARS-CoV-2 pandemic right now because it's not as severe. Fewer people are getting sick with it. So why are we still worried about pandemics? So although the threat has lessened from SARS-CoV-2, the virus in its current form now is less severe than it was in 2019 and 2020. The SARS-CoV-2 virus is still out there, it's still mutating, it's still evolving. So there exists the possibility for it to come back in a different form that is kind of harmful to us, and we'd like to not have to deal with that. Furthermore, even if SARS-CoV-2 were to completely disappear tomorrow, there are still so many bacterial and viral pathogens that are out there that we are very concerned could launch another pandemic. So this is an image taken from the Spillover website. This is a group that said, hey, let's look at all the viruses that we know that are out there infecting wildlife, and let's get a risk scoring going so that we can rank which of these viruses we are most worried about spilling over from wildlife into the human population and causing a pandemic. You will note that SARS-CoV-2 is on this list, and that's because SARS-CoV-2 is happily circulating amongst the deer populations of the world right now, and we're very concerned that it could spill over from deer back into humans in a new, changed, scarier form. It's great. But there are plenty of other viruses that are on this list as well that we are very concerned could spill over into humans and cause a pandemic. So we need to have tools available now for handling these things before we have another global pandemic. So our group had a couple of questions. Number one, can we identify pandemic threats before people get sick? Can we figure out that this virus that popped up in some faraway place is going to be a problem before it becomes and blows up into a big problem? And can we make vaccines and treatments for these threats before people start getting really, really sick, before it becomes a global pandemic? So obviously my group thinks that the answer to these questions is yes, we absolutely can do these things. Otherwise I wouldn't be here, talking to you guys about them. It would be a very boring presentation. So what my group is doing is using artificial intelligence and large language models to work on combating these threats. So what is a large language model? A large language model is a computer model that is trained to understand written text like a human being does. They are called large for a couple of reasons. One is that they take truly extraordinarily huge amounts of data in order to train really, really good models. So language models trained for human text, we're talking hundreds of terabytes of information potentially to train these things. They are also huge in that they're physically big models. They have potentially hundreds of billions of trainable parts within the model. We call these parts parameters. So they're very, very large language models. Language models are everywhere. I guarantee you that if today is the first time that you have heard the words language model, this is not the first time that you have interacted with a language model. Any of you guys heard of a little something called ChatGPT? Yeah, a few hands going up there. So Chat GPT is not just a thing for, doing your homework for you or automating text generation. It is a language model, but language models are also out there doing things like spam detection, language translation, summarization of scientific articles and news articles, code generation, sentiment analysis. This is a really fun one. We can tell what the public thinks about something by finding out how angry they are about it on Twitter. Toxic language detection, which is also really useful when you're interacting with Twitter. Basically, if it involves language analysis or generation of any sort, a large language model is probably involved in that process. You are interacting with them every single time you are on the internet. They are incredibly useful. There are lots and lots and lots and lots of different types of language models out there. So here's ChatGPT off in this corner. You will note that there's a whole bunch of other stuff on this chart that's not ChatGPT. All of these guys are language models. ChatGPT is honestly just one of the latest ones that built off of a bunch of other GPT models. So all of these models, as I said, they're language models. These guys that are in orange and blue and red, though, they are a specific type of language model. They are built off of something called a transformer. Not that kind of transformer. They have absolutely nothing to do with Autobots. They have absolutely nothing to do with electrical transformers. They're called transformers because they transform input into output through a structure called a transformer. I'm not going to get really into the nitty-gritty of how transformers work because there is not enough time in the day for me to cover that. But the short version, the punchline that you should take away from this is that transformers learn relationships between words and concepts through something called a self-attention mechanism. So basically, when you give the model a whole bunch of words, it learns the relationships between these words. And those relationships can include things like how parts of speech interact with each other, how concepts are related to each other. It's a very, very powerful method of learning. And that's why, if we go back to this chart here, you'll note that all of these non-transformer things pretty much just stopped in 2018, 2019, which is when transformers first appeared on the scene. They were so powerful, they fundamentally changed the landscape of language modeling. And so now basically everyone uses transformers. All right, so how do computers learn human languages? Because we can't just give a sentence to a computer and then the computer does stuff with that. It takes a lot of learning. Just like if you speak words to a baby, the baby's not giving you Shakespeare back anytime soon. We have to teach the model to read and understand a language, and we call this process pre-training. There are a lot of different methods of pre-training out there, but one of the most common ones, my animations are a little screwed up here. One of the most common ones is to take a sentence, to mask one of the words in that sentence and teach the model, the computer model, to predict what that missing word is. So this is called masked language modeling, and it's a really, really powerful way of training the computers to understand human speech. We next fine-tune the model on a task-specific language. So for example, I work with a lot of influenza data because that's another virus that we're really concerned about. And influenza data comes with metadata files from something called GenBank. So these GenBank files, they have a lot of information, including in some cases, what organism the influenza was cultured in a laboratory. And that's important for us to know because it can have implications for how influenza is changing and adapting to things. The unfortunate thing is that up until a few years ago, nobody really paid attention to that. And so they kind of just buried it in these metadata files without labeling it nicely. There are 600,000 of these metadata files that I have to go through to find this what's called passaging information. And I don't have time for that. So I trained a language model to read these GenBank files and pull out that passaging information for me. So my fine-tuning process then was taking these GenBank files and doing the exact same thing, this masked language modeling task. And then I gave it a much smaller data set that included that passaging information and labeled so that my computer model can learn, okay, so in answer to the question of what is this influenza sequence's passaging information, I need to pull out that corresponding information from the GenBank file. And this works shockingly well. I get like 99% accuracy doing this. It's very, very nice. I don't have to read 600,000 GenBank files. But so this whole process, this applies to basically any language model that we're training. We first do this pre-training where we teach it to understand a language. We then fine-tune the model on the task-specific language, and then we finally do a specific task with that model. Okay, so we covered then the general process of how computers learn, but like, how do we take words and have a computer actually understand what we're doing with them? So computers do not like words and letters. They just don't. You can throw words at them all day. They don't really like to do anything with that. They really, really like numbers and math. So in order to use computer models to do anything with language, we first have to turn language into math. And we do this through the use of something called an embedding space. So right here, you can think of this exactly as a map. This is a 2D map of a space. And we call this space the embedding space or the latent space. And we can actually use computers to map words to coordinates within this space. So we've got our own little country over here, and we've got another little country over here. Each of these dots represents a word or a concept. So the coordinates of these words within that latent space, this is called an embedding, and those coordinates are numbers. Our computer models can do math upon those embeddings and coordinates. And they do math really well upon those embeddings and coordinates, actually. So the really, really cool thing about embeddings and latent spaces is that we expect that things that are related to each other are going to be nearby each other on this map in this latent space. So here I've got a few words that are mapped onto this latent space. We've got an apple and a pear, king, queen, man, woman. You'll note that these guys are clustered down here away from these guys. These guys are plants up here. This is a human concept down here. So those different concepts are going to localize to different areas of the map. So this is really, really important to how language models operate. They're able to do math upon these related concepts. All right, so at this point, I've spent like 15 minutes talking about things that are not really biology related. I've been talking about how we use math to make these cool language models where we can do tasks with the English language. All right, so where does biology come in? So to explain how the biology relates here, we're going to do a really, really quick down and dirty biology lesson. All right, who here has heard of DNA? All right, good, most of you. For those of you who didn't raise your hands, on the off chance that you actually don't know what DNA is and didn't just want to raise your hand, DNA are blueprints. They hang out in almost every cell of your body, and your cells use them to make things. DNA blueprints are kind of important. We don't want them to get damaged by things like radiation or chemicals. So they hang out in the middle of your cells in a structure called the nucleus, where they are protected. Where your body still needs to use them to make stuff. So it makes copies of the DNA called RNA. And those copies can then be taken out to your cell's factories where they are used as the blueprints to manufacture stuff. And that stuff is proteins. So proteins do things like make up your fingernails and make up your skin. They make up your muscles. They do all sorts of structural things. They also do things, and unfortunately this is a little bit blocked here. They do things like carry oxygen to your cells and help digest your food and protect you from diseases. So proteins do a whole bunch of things. Thank you. So this is a human hemoglobin protein. So that's what's carrying oxygen to your muscles. So here's the thing about DNA and RNA and proteins. These guys are all sequences. So DNA is a set of what are called bases, adenine, guanine, thymine, cytosine, that are all hanging out in these long DNA strands. And this forms a code. And this code is eventually translated into a sequence of amino acids. There are 20 of them. So there are 4 letters in this alphabet. There are 20 letters in this alphabet. These are codes. These are languages. We can use language models that were created for understanding English and Chinese and French. we can use these to understand biological sequences. If it can be turned into a sequence, we can use large language models on them. And indeed, we don't just, map human words and concepts to these latent spaces. We can map proteins and DNA sequences and RNA sequences to these structures and areas as well. So here we have a human hemoglobin hanging out up here, but we now have a country on this map that's corresponding to viral proteins. This is an RSV fusion protein. This is influenza hemagglutinin. This is SARS-CoV-2 spike protein. These are all in the same area because these are all proteins that allow a virus to grab onto a human cell and infect that human cell. So they are all structurally and functionally related. They have their own country in the map. All right, so our group, finally, I'm talking about our research and what we did with this. Our group trained some massive large language models. We trained them on 110 million DNA sequences taken from this Bacterial and Viral Bioinformatics Resource Center, or BVBRC. We trained several models. The two largest have 2.5 billion and 25 billion trainable parameters, so they are very large. We have trouble getting them to fit on our supercomputer chips. On the right here is another map. This is exactly the same kind of map that I was showing you on the previous slides, except the dots on this map correspond to DNA sequences and not words or concepts. So this model isn't really taught anything about these DNA sequences, but it still clusters similar things together. So this is coloring these sequences by their sequence length. You can see that we have these nice, really clear clusters that correspond to sequence lengths. So it's learning some properties about these sequences and putting them together in our latent space such that things that have similar sequence lengths are in the same area. We do the same thing with GC content. So that's how many guanines and cytosines are in these DNA sequences. We see these nice gradients where, again, higher GC and lower GC contents are all hanging out by each other. So that was a great sign to us that these models were actually understanding and comprehending the DNA sequences that we were giving them. This was just another fun figure that's painted to show that they also cluster based on their functionality. So these light blue sequences that you can see, these are all DNA sequences that make machines that have to do with metabolism, so supplying your body with energy. We got a nice little island over here that's stress response, which is pretty cool. And we've, got these really, really nice groupings of all of these functionally related DNA sequences. So again, our model is learning things about these sequences that we didn't explicitly teach it. So cool, we have these massive, massive language models. What are we going to do with them? Well, we decided that we were going to take these models and we were going to fine tune them on some SARS-CoV-2 data. So we made a data set that included SARS-CoV-2 genome sequences that were sampled up to January of 2021. And we trained our models, we fine-tuned them on this data. So first we taught our models how to speak DNA. Now we're teaching them how to specifically speak SARS-CoV-2 DNA. So this was about 1.5 million genome sequences that we gave our models. So just a refresher, prior to 2021, really all we were seeing was Alpha and Delta. After 2021, we started seeing Omicron, a whole bunch of different variants were popping up. But our model has only seen this stuff. So what was really cool, we trained the model, we fine-tuned it on these SARS-CoV-2 genome sequences, and then we said, hey, how well is this model able to map new sequences into this latent space? So we took another data set, and this data set does include data up to 2022. So it includes our Omicron sequences and all those other fun variants. And we are able to very, very nicely cluster these 16,000 new genome sequences into their own little lands based upon what variant they belong to. We didn't explicitly tell the model, hey, this is an Omicron variant or this is an Alpha variant. It picks up on its own, and it puts these things in their own little areas of the latent space. So we have our Omicron countries and our Alpha countries and our Delta countries. Model does that all automatically, which is really, really cool. So here's a fun question. What happens if we take the coordinates, of like right here or something, and we, backtrack? So let's take those coordinates and let's transform that into a SARS-CoV-2 sequence. What would happen then? So we can do this, actually. We can sample different areas where, we don't necessarily have any sequence data. And we can transform that back into a SARS-CoV-2 sequence. We use a special kind of computer model called hierarchical diffusion models. We're not going to get too deep in the weeds on that, but punchline, we can sample all throughout this latent space, not just where SARS-CoV-2 genomes that we've seen in the past are. And we can then use those transformed sampled sequences to start forecasting where SARS-CoV-2 might be headed next. So we did that. We sampled 1,000 different points within that space, kind of as a proof of concept thing. And then I mapped those genomes that had been sampled from the latent space onto actual SARS-CoV-2 genome sequences. So this is a phylogeny. That's a tree of life here. All of these sequences in blue are things that our model generated. These don't actually exist out in the wild that we have measured yet. But we were still seeing these popping up on, they're mapping to Delta, they're mapping to Omicron, they're mapping to all of these other variants that we've seen out there. So these are all biologically plausible SARS-CoV-2 genomes that we haven't yet seen in the wild, but theoretically could exist in the wild. So it gives us an idea of where SARS-CoV-2 could be changing next. So another really, really cool thing, getting back to the transformers that I was talking about, remember how I said that they can learn to pay attention to different parts of a sentence? Well, our transformer models can do the exact same thing with a SARS-CoV-2 genome. So this is a diagram showing the SARS-CoV-2 genome here, and these orange lines represent different parts of the SARS-CoV-2 genome that are being paid attention to each other via our transformers. So these attachments, these relationships suggest parts of the SARS-CoV-2 genome that change in conjunction with each other. So we can start using this kind of information to say, all right, we've seen these changes in the SARS-CoV-2 genome. We're kind of expecting that now this other part of the SARS-CoV-2 genome is going to change as well. And if it's changing in a part like the spike protein that our vaccines target, that gives us a head start on looking at how that might change and how we might need to adapt our vaccines to change as well. Okay, so like I said, we trained this model on data up to 2021, and then we kind of looked at 2022 data. So my boss came to me a few weeks ago and said, hey, can you pull out those 1,000 genomes that we sampled last year? And can you just see if any of them look like the stuff that we have circulating right now? So we have a couple of variants that we're concerned about right now, EG.5 and XBB.1.15. And so I said, sure, boss, I will do this thing that you have asked me to because you are my boss. And so I went and I looked at these 1,000 genomes, and sure enough, we had some of these genomes that, again, the only data they've seen was 2 years removed from what we're doing right now with SARS-CoV-2. And we still had genomes that looked pretty darn similar to what we had going on in 2023. And unfortunately, the transparency didn't work so hot on this image right here. But these are, this is the SARS-CoV-2 spike protein, and I have mapped to it in orange and red those places where our generated sequences match up with the changes that EG5 and XBB.1.15 have from the original SARS-CoV-2 genome. So again, something that we generated back in 2021 looks an awful lot like what we're seeing in 2023. And we only had to generate 1,000 genomes to get these kinds of results, which was kind of surprising to us. We thought we would need to generate a lot more, but the really, really awesome thing about these models is that they can see based on just, that 1 year of data, how SARS-CoV-2 changed over that 1 year. They can start picking up on how SARS-CoV-2 is more likely to change in the future. Can you click on the thingy? There we go. Thank you. Okay. So what I have right here is another one of our generated sequences. We took several of these SARS-CoV-2 genomes, we extracted the spike protein sequence, and then we threw them on our supercomputers and said, hey, supercomputers, can you take this sequence and turn it into a 3D structure for us, pretty please? And so we were then able to examine, using these 3D simulations and a lot of supercomputing time, we were able to examine how these SARS-CoV-2 generated spike proteins are going to bind to a human ACE2 receptor. ACE2 receptors are the thing that SARS-CoV-2 grabs onto in order to infect a cell. So if a spike protein comes along and it's able to grab more tightly onto ACE2 than other spike proteins in the past, that's a good sign that that SARS-CoV-2 sequence is going to be more infective than what we've seen in the past. And it could be, a little more dangerous for us. So in blue, we have our generated sequence here. And in yellow on this thing, it's a little hard to see, but in yellow, we have point mutations, how our model predicted that this SARS-CoV-2 genome is going to change. And these little point mutations, we can actually calculate how tightly that will cause this new SARS-CoV-2 spike to bind onto our ACE2 receptors. So these are graphs that are showing that generated sequences in relation to how tightly other SARS-CoV-2 variants grab onto things. So things that are above this line here, that means that our generated sequence is grabbing on less tightly than what we're seeing in nature. But things that are below that line, that's an indicator that our generated sequence is grabbing on more tightly than what's out there in nature. And so that's, we can tell then that if we see something that looks like that sequence in nature, oh boy, that's a thing that's going to be able to grab harder onto our ACE2 receptors and could potentially infect more people, could potentially become one of those variants of concern that you're frequently hearing about. So that was pretty darn cool to see that our generated sequences are in fact showing, these are potentially more fit SARS-CoV-2 genomes. So this could be where we're kind of headed with SARS-CoV-2. So most of this work that I've shown you is in a paper that is going to be published very shortly. These are called GenSLEMs, genome scale language models. And we were able to reveal some SARS-CoV-2 evolutionary dynamics, which was pretty fun. This work was actually awarded the ACM Gordon Bell Special Prize for HPC-based COVID-19 research. The ACM Gordon Bell Prize is sometimes called the Nobel Prize of supercomputing. Not to toot our own horn in there. It's not actually the Nobel Prize of supercomputing. This is worth $10,000. The Nobel Prize is worth $1 million. Also, usually only 1 to 3 people get the Nobel Prize. This prize was spread out amongst 30 people. I got $300. It was great. But ACM Gordon Bell Special Prize, it was pretty cool and quite the feather in our caps for this work. All right, so just to recap what we've gone over today. So number one, we can use language models on biological sequence data. So if it is a sequence, you can use large language models on it. We trained a large language model on 110 million DNA sequences. We then fine-tuned on 1.5 million SARS-CoV-2 genomes. And then we are able to build downstream tasks on those models to try to sample likely SARS-CoV-2 genome sequences. We can do a lot of other stuff with those models as well. I just, don't have enough time to talk about all the things we're doing with those models right now. So with just these 1,000 sampled genomes, we are able to capture SARS-CoV-2 sequence dynamics sometimes years in advance of when they are occurring. So this is giving us the possibility of being able to start analyzing SARS-CoV-2 sequences now and figuring out, okay, when people are out there sequencing SARS-CoV-2 genomes from the general public, if we start seeing variants that look like this, that's maybe a sign for alarm that we need to ramp up our response in those areas. So all of this is so that we can start preparing now and being proactive in our response to pandemics instead of being reactive in our responses. So as I mentioned, there are a lot of things that we can do with those models, and a lot of that work is ongoing right now. But just to give you kind of, some of the highlights of the stuff that we're working on. We're working on models for more poorly sequenced diseases and viruses that are out there. We are particularly concerned about Lassa mammarina virus and Nipah virus. You may have heard of Nipah. It is currently causing problems in India and Bangladesh. So we're, very concerned about that potential for a pandemic. We're also working on models for bacterial agents. Those are more tricky because bacteria is a lot bigger than viruses. SARS-CoV-2 has 10,000 letters, or it has 30,000 letters in its code. We represent it as 10,000 letters, but SARS-CoV-2 has 30,000 bases in its genome. E. coli, a very common bacteria that can cause illness, has about 4.5 million bases in its genome sequence. So it can, be tricky working with things that are that large. We are also working on large language models to identify the parts of viruses that we should be targeting for vaccines and other treatments. In fact, I am running a model, I'm training a model for that right now, actually. So all of these things we are working on through the use of artificial intelligence and large language models. So this is not the end of the talk. I am going to talk about a few more things, but I just want to acknowledge some people who were involved in this research. And I also really, really want to thank these people. They have thrown literally millions of dollars at this project. We are extremely grateful for all of that money. It pays my salary. But there are, as I said, there were a ton of people that were all involved in that GenSlim work. And a lot of them, they're just absolutely brilliant scientists, including some graduate students and a couple of undergrads as well were involved in this. So big thanks to all of those people for making this work possible. All right, so how did I come to be an assistant computational scientist at Argonne National Laboratory? So I don't think of myself as an assistant computational scientist. I think of myself as a computational biologist. So my journey starts actually in Illinois in a town called Aurora that you may have heard of because it's only about an hour or so away from here, assuming that there's not really disgusting traffic. There's always really disgusting traffic. But I went to Waubonsee Valley High School in Aurora. I actually grew up just about 45 minutes away from Argonne National Laboratory. My dad actually worked at the credit union while I was growing up there. I then got my undergraduate degree at Augustana College in Rock Island, Illinois. That degree is a bachelor's in biochemistry. And then I went a little farther west along I-80 and ended up at Iowa State University, where I got my PhD in bioinformatics and computational biology. I was really sick of being so far away from all my family that's located in Chicago. So after I graduated, I came back here and I got a job at Argonne National Laboratory, working not actually on this research. We were working on a different project to put self-destruct buttons in bacteria. It was super fun. So how did I get to computational biology? So unfortunately, the animations didn't work on this because it was a lot more dramatic and funny when I had them, but bear with me here. So in 4th grade, I knew that I was going to be a scientist. I was that weird little kid that had a microscope at home and would go around collecting pond water and looking at paramecium under the microscope and whatnot. In 9th grade, I knew I was going to be a geneticist. We had our honors bio unit on DNA, and I thought it was the coolest thing ever. I still kind of think it's the coolest thing ever. But so I knew I was going to be a geneticist. So throughout high school, I was taking classes towards that goal. So as a freshman in college, I decided that rather than get a straight up biology degree, I wanted to get a biochemistry degree and then use that to work on becoming a geneticist and solving hereditary illness and everything. So as a sophomore in college, and actually before I was a sophomore in college and also up until today, I play video games. I play lots and lots of video games. So as a sophomore in college, the big game that was going on right then was Minecraft. I started playing Minecraft version 1.7. So if that gives you an idea how old I am. So as a sophomore in college, though, I was enjoying Minecraft and I was like, all right, I'm going to learn how to code so that I can mod Minecraft. So I started taking computer science classes in college explicitly so that I could learn to mod Minecraft. So fun fact, I don't actually know how to mod Minecraft today, but I do have a degree in computational biology, so I guess it worked out. So the summer of my senior year in college, I had to do a biochemistry lab project, kind of like as a capstone thing. And so I spent an entire summer, a summer of my life that I will never get back, doing the exact same experiment over and over and over and over and over again, expecting a different outcome. And I realized that I don't actually like doing biochemistry lab work. I actually kind of hate it. But I was a senior in college and I really didn't want to go to my parents and say, hey, mom and dad, thank you for paying for 4 years of college, but have I got some news for you? So instead of throwing out everything that I did, I kept at it under the logic that maybe if I just keep doing this, I'll learn to like it again. But fortunately for me, fall of my senior year, we had a molecular genetics course that we had to take, which I did enjoy. But the part of it that I enjoyed the most, we did a bioinformatics module, and I had a wonderful time using computer science to analyze the biological data. And I didn't have to sit in a lab doing the same experiment over and over and over again, wondering why it didn't work this time. And so I liked that so much. And also all of my classmates hated it so much that I thought that's probably a field that has some career opportunities for me. So winter of that year, I applied to Iowa State University's Bioinformatics and Computational Biology program, and I was accepted, and the rest is history. But I just want to emphasize that the thing that I have my PhD in, I hadn't heard of bioinformatics until we did that unit. So I hadn't heard of the thing that I do as a living until about 6 months before I went to grad school for it. So, be open to new opportunities. They can hit you fast. So fall of 2019, after I finished getting my PhD, I started working as a postdoc at Argonne National Laboratory. And actually, October 5th, I transitioned from being a postdoc at Argonne to being an assistant computational scientist, which is kind of Argonne's version of an assistant professor. All right, so if you too are interested in computational biology, if you thought any of what I talked about is kind of interesting today, you will need to do some math. I am now at the point in my career, I don't like math, but I'm at the point where I can get computers to do all the math for me, which I really, it's a good system. You do have to know statistics, and that is different than math. All of the computer models, everything that I showed you, that all works off of statistics. Large language models, just applied statistics. Computer science is also important, and you'll note that this is different than computer programming. So you actually need to kind of understand the hardware and machinery and how computers operate in order to get your models to train properly. We have a bunch of people who for months now, they've just been fiddling with their code to get it to run as fast as possible on the hardware systems that we use. Computer programming, of course, good to know. Even if you're not going to do computational biology, if you're not going to do computational anything, computer programming is one of the most versatile, broadly applicable skills that you can learn. So I highly recommend it regardless of what you're going to do out there. Like, even if you're just working like a desk corporate job, like there are people out there who have used computer programming to completely automate their jobs. So they're paid to be there on their phones because they automated everything already. Don't tell your bosses that you automated your job. Pro tip. You also, of course, need to know at least a little biology if you're going to be a computational biologist. But actually, it's a lot easier to teach computer scientists the biology they need than to teach biologists the computer science they need to do computational biology. I also want to just share with you all that Argonne has research opportunities. So if you are at all interested in STEMmy kinds of things, we have undergraduate internships. We have paid undergraduate internships. The Science Undergraduate Laboratory Internship Program, or SULI, is open right now, and I believe it closes on January 9th. This is a program where you get to go to Department of Energy labs and do actual research. Like one of the people that's on that Genslim, she got $300 too. She's an undergrad at U Chicago, and not exactly this program, but basically this program. She spent 2 summers with us there. So SULI internships, they're really, really useful. Also, ANL.gov undergraduate programs. We have other undergraduate programs. You should check them out. They're really, really cool. All right, and with that, I will take any questions you guys have. And if you don't want to ask a question here, think of something later, this is my email address. I am happy to answer any questions that you guys have. Thank you. Any questions? Yeah.
Congratulations on your award.
Thank you.
Thank you for the work. I noticed the NIH is one of the sponsors.
So we don't have a ton of involvement in SARS-CoV-2 vaccines. We do actually have some involvement in influenza and picking what influenza we're going to vaccinate on any particular given day. I will say that a lot of those conversations happen several levels above my assistant computational scientist pay grade. But I was in a meeting that had members of the CDC a couple of weeks ago. And so we are certainly having those conversations, or rather my boss's boss is having those conversations. But we do think, like, we give them this modeling data. We have a lot of people at Argonne who were analyzing just tons of SARS-CoV-2 data during the pandemic. So this may be unpopular because of how they were implemented, but a lot of the COVID restrictions and recommendations that were implemented in the city of Chicago were implemented because of models that Argonne was running. And those models did actually work really, really well. Whether or not the implementation of them was good or not is a different question. But so we are certainly sharing data, and we have like people in the building on a monthly basis that work for those groups. So we have those conversations a lot. That's a great question. Yes.
So the thing. About the.
Okay, so I am frequently asked, like, okay, so when is the AI gonna become self-aware, and we're gonna have Skynet, and we're gonna have, Judgment Day and Armageddon and whatnot. I am not even remotely concerned about that happening. Honestly, if you look at the AI that we have right now, like ChatGPT looks smart. ChatGPT is so dumb. Oh, it's so dumb. Yeah, I'm not worried about those things. What I'm worried about is how human beings will use these computational tools. We've actually seen in the past, like there have been models that are created to try to predict crime. How many of you guys have seen Minority Report? Okay, like 2 of us. All right, so I won't make the Minority Report reference. What I will say is that human beings tried to treat those models predicting where crime was going to happen as fact instead of a prediction. And so they ended up causing a whole bunch of harm to their communities because people hadn't been committing those crimes. The models just thought that they were, and how people used them was improper. That is what I am worried about, is how people will interact with these models. We're actually, like deepfake technology, that's fairly terrifying to me. I'm not terrified that we have the capacity. I'm terrified of how people will use it. Same thing with some of the image generation stuff that we got going on. Yeah, all of these things, they're issues with how people interact with the technology rather than the technology themselves. So like, I think that I've done a decent job arguing here that we can use this technology for really, really cool purposes that is genuinely helpful and beneficial to people. But you can also turn around and, use any of this technology for nefarious purposes. So yeah, it's a concern, but I'm not concerned about Terminator happening, if that makes sense. Any other questions? Yes. Yes, so these models, they can be used for prediction. Whether or not they can be used for good predictions right this second is a matter of some debate. But we're very confident that as we get more data, these predictions are going to get much better. And we're actively, yes, using these to try to forecast where NEPA and LASA are going to go. We are really, really concerned about NEPA and LASA, guys. Other questions? Not necessarily about research, but anything in general. Yes.
I knew that I won't have a supercomputer, but
so to train those models, to train on the 110 million sequences, if we get to use every single node on our supercomputer, it takes about 2 days. If we don't get to use a supercomputer for this, we just flat out can't train these models because our computer would die of old age before the model finished running. Supercomputers are very, very important for large language models and AI. So yeah, it takes quite a while if you don't have a supercomputer.
So, I pray, so we have our, with it, how many people like?
So this is actually the meeting that I was at 2 weeks ago that included people from the CDC. This was one of the conversations that we were having is obviously we want to be able to forecast and be able to, proactively go after things rather than react. But even if we have the best systems in the world, we can always be blindsided by something that we never saw coming. Like some bat somewhere in a cave that has never once seen a human being in its existence has some weird virus, and it just happens to stumble upon somebody who is walking out in the forest that day. And oh no, we have something that we have never seen before. And so that is some of the calculus, is that if in that scenario, it is probably inevitable that, yeah, people will get sick and people will die before we have the opportunity to respond to those things. So our goal is, and our hope is, that if we can model as many viruses that are out there right now and kind of get the dynamics of how those viruses change over time, that will give us a better idea of what all is out there. And so our models will hopefully be able to better handle exactly that kind of scenario. The project that I am currently funded off of, their goal is actually that within 100 days of disease X, whatever new creepy thing pops up, within 100 days, we want to have a vaccine for it. And I think that we do stand a shot of actually achieving that goal, but it involves a lot more sequencing information than we currently have included in these models. But again, with the supercomputers, we think that we can, within 2 days or so, have new models trained on that information. Great question.
Yeah. So now you have a model. So if
we don't have to retrain the 110 million part. So we can take that model, we call it a foundation model because it can act as the foundation for lots of other things downstream. So we can take that understanding of DNA sequences, and then we can fine tune on different types of genomes. So the family that of viruses that Nipah virus comes from, paramyxoviridae, we can make a language model then built on top of our 110 million model that understands paramyxoviridae DNA sequences, for example. Yeah, saves a lot of time, actually. All right. If nobody has any other questions, again, if you think of something, my email's up there, happy to answer. But thank you for your time and attention.